when we talk about mobilization of phagocytes, what I mean by that is if you have, say, an infection, okay, something that is initiating an inflammatory response, you are first going to see an increased number of neutrophils at the injured site or the infected site. Now, neutrophils, although being dominant one at the start, macrophages will predominate at the end. Okay, they're the ones that's going to be doing the heavy lifting. Okay, but neutrophils are the first ones to go there. Now, if inflammation is initiated by a pathogen, instead of just neutrophils and macrophages, we are also going to have another group of plasma proteins that will arrive on scene called complement. You've heard me say this now the third time, and the next video or two we'll talk about complement. We can also have a variety of other lymphocytes and antibodies from your adaptive immune system to then come to the site as well to start adaptive immunity. So let's look at what the different steps of mobilizing phagocytes on this one. Okay, the first step is going to be what's called leukocytosis. Okay, what leukocytosis, leuko white blood cell cytosis is a lot of. We are basically, chemicals are being released, tells your bone marrow, red bone marrow, to, hey, make more neutrophils. Okay, we've got an infection, we need more frontline defense, make neutrophils. Those neutrophils go to the area of infection, and an increased number of white blood cells, and in this case, in one area, is called leukocytosis. Okay, so that's something we want to know, where we increase the number of white blood cells in the area. The second one, and we're talking neutrophils in this case. The second one we're going to be calling is margination. Okay, and marginization, and I've got a picture here in just a second. We'll go through this again. Picture a capillary, okay? And in the capillary, you have your red blood cells, white blood cells, all that kind of stuff. Now, we want those neutrophils to come out of the capillary and go to the infected area. Okay, so now we're talking about how the process is where that neutrophil comes out. When you have an injury, an infection or something, and the injured endothelium, got a capillary, that inner lining, the endothelium, begins to sprout or grow out little hooks, if you will, okay? And those are called cell adhesion molecules. So margination, this is all going to be underneath marginization, okay? So we're going to have cell adhesion molecules, and you'll always see that abbreviated CAM, okay, or CAMs. And that is, they're little sprouts from the endothelial lining of the capillary or the injured vessel. And what those are doing, it's basically telling the neutrophils, hey, over here, this is where you want to go. This is the area, okay? So the neutrophils kind of know when they are located, or they're located at the position of the problem, okay? So what happens at this point is the neutrophils migrate to, if we got the capillary here, and you got a whole bunch of neutrophils in here, they migrate to the endothelial wall. So they're going from the center or wherever they're at, and they migrate to the sides. And we know that the velocity is slower in that area, and the neutrophils slow down, and when they're on the endothelial lining, they begin to roll or tumble. Think like a tumbleweed, all right? They're just rolling slowly. And the neutrophil also has CAMs on its plasma membrane. And when the CAMs meet up, they stick. So they lock together. So the neutrophil is at the site that it's supposed to be. And then it begins the process of leaving the blood vessel, and that's called diapedesis, which is, let me get that spelt right, Diapedsis, I think. Yep. And diapedesis is basically migrating out of the blood vessel. Okay, so diapedesis, it's walking out of the blood vessel. Okay, and now once we have the neutrophil outside of the blood vessel, it now needs to go to the site of injury, or to the site of where the bacteria or something is, and that's going to be called chemotaxis. And chemotaxis is basically the neutrophils acting like a bloodhound. All right. Anybody who's got a dog, if you hide a treat, and you're playing around with your dog and you hide a treat somewhere, their nose goes up in the air and they try to find it. Okay. And it's kind of fun watching them going around the living room and they're tracking down the smell. Well, neutrophils are just that. Think of a neutrophil or a macrophage or any other cell that does this, phagocytic cell. Think of it as a big nose, and it's tracking down the bacteria. Now, the bacteria, the injured tissue, releases chemicals that the neutrophil is following. Okay, it's following a trail. And that's called chemotaxis. Now, the neutrophils are the first things on site, but they eat one bacteria, they die. They eat another one, eats a bacteria, it dies. Macrophages are your predominant pig, all right? They eat everything. They just love eating, so they can eat a whole bunch of stuff. But macrophages originate from monocytes, and monocytes aren't a good phagocyte, or is not a good for phagocytosis. Monocytes leaving the bloodstream gets into the interstitial fluid, and it begins to get larger. And when it gets larger, it's making more lysosomes within it. Now remember, lysosomes contain some of like those digestive enzymes. So it starts to having a lot more of those, and then that follows chemotaxis as well. So let's look at this image that I put here. Here we got our capillary, or some kind of a blood vessel here. And these are just basic, don't worry about the blurriness of this, it's blurry on my end. These are, let's say, your neutrophils, and your site of injury is way over here, okay? So what happens is that neutrophil migrates to the endothelial lining, and it starts to tumble. So you can see that this is rolling. You can see how it's rolling around. But what you don't see on here is, and I'm going to do this in green so you can see it, is the endothelial lining has these little, and I'm just drawing them in, and those green things are called cams, okay? So, all right. But you also have them on the neutrophil. So when that neutrophil is doing its best imitation of a tumbleweed, okay, once those cams attach, then the neutrophil's in its spot where it wants to be. The neutrophil flattens out. You can kind of see we have a nice round structure here, but now it's flattening out. You can see how this is getting even more flat. And here we see it wanting to come out. Coming out, this is going to be the diapedesis area. So that's diapedesis. And you've got your injured tissue, you've got your bacteria in this area, and what it does here is it uses its big nose and it tracks down. Think of your dog sniffing around for that treat, and it's finding the injured tissue, the damaged tissue, the bacteria, whatever it is, and it will phagocytize that. So that's phagocytosis. Next thing we'll talk about is interferon.